The overall goal of the following experiment is to quantify and size profile extracellular vesicles using tunable resistive pulse sensing. The standard approach is to compare current blockade measurements taken from purified extracellular vesicles with those taken from polystyrene bead standards. A second approach to characterizing extracellular vesicles is to spike the sample with polystyrene beads, which is used when working with complex samples such as unpurified extracellular vesicles in cell culture medium. The resulting data characterizes the size and number of the extracellular vesicles, whether they are purified or unpurified.
The main advantage of this technique over existing methods like Western blotting or flow cytometry of extracellular vesicles bound to latex beads is that this method characterizes particles directly in biological fluids without the need for vesicle isolation or labeling.
Generally, individuals new to the method will struggle because extracellular vesicles are heterogeneous in size. When trying to detect the smaller vesicles, larger vesicles may clog the nanopore. We have added some practical tips and tricks to the protocol to quickly return to workable conditions.
We tried to reduce the production of extracellular vesicles and needed a method to quantify the concentration of vesicles in cell culture supernatant. In search for methods to characterize nanosized particles, we came across TRPS and modified its protocols to be more suitable for characterization of vesicles in cell culture supernatants and other biological fluids.
Begin by connecting the TRPS instrument to a computer with the Eyes-on Control Suite software installed on it. Be sure to minimize electrical